I hope you can hear me okay. And I think I can see my slides. So I'm very, very grateful to be here with you all. Um, I feel very, very disappointed that I cannot be here with you in person. I actually 我也会讲国语，所以我真的觉得我很希望我可以跟你们在一起。But again, sadly, I'm in California having to deal with a medical emergency. But I'm grateful to Naomi and very grateful to the Venerable Yifa for organizing this very, very wonderful and important event. I wanted to start by also acknowledging my parents and telling a little bit of a personal story. So this is my mother and father. They both actually went to Taida. So they spent a lot of time growing up in Taiwan. And both of my parents ended up in the greater Boston area. And part of my own background and heritage, I'm actually a direct descendant of Confucius, of Kongzi. So I appreciated the opening remarks about talking about Confucianism and Buddhism. And this is part of my own heritage. I'm a 75th generation direct descendant of Confucius. This is my family, and my father, again, was a professor at MIT. My mother was a professor at UMass Boston. And my father actually explored research in what's called electromagnetic wave theory. So 5 fundamental equations that govern all electromagnetic phenomenon in the universe. And he was very interested not only in electromagnetics, but also in Chinese philosophy and literature and history. And so one of his favorite texts was the I Ching. And in his late life, he was very, very interested in this intersection between science, electromagnetism, electromagnetics, and also philosophy. And so for me, this is also part of my own heritage is this exploration of science, technology, and thinking about spirituality in this context, in particular Buddhism. And so I am from the Media Lab at MIT. I would so love to welcome any of you to come visit here in Cambridge and in the Boston area. And at the Media Lab, we explore science, engineering, also art and design. And the group that I lead is called the Community Biotechnology Initiative. And the research area that I explore is called synthetic biology. And we've learned a bunch about synthetic biology the past few days. I'm very grateful to Dr. Gao for his wonderful presentation yesterday, introducing some of the concepts in synthetic biology. And I'll return to synthetic biology, but I think for me, again, I'm not able to join in person, but I'm grateful to Naomi and to the live stream. I was able to watch the events yesterday. And it felt like there were a number of very important questions that you all were discussing that have very significant implications when we think about synthetic biology and synthetic life. So one question is, what entities do we consider to be living? How do we define what is alive? What types of entities can we define as being sentient? And also, what types of entities can we define as having consciousness? And I think this is almost a technical type of a conversation and a philosophical debate about what are the characteristics for each of these questions. And if we can decide upon these characteristics, then I think some of the questions that connect to Buddhism, and I should mention again, I'm not a Buddhist scholar. I'm a scientist and technologist, but I'm very interested and inspired by Buddhism. But questions like what beings suffer, and I use the word entity versus being, entity being different types of objects or artifacts that are part of the universe, but beings having entities that actually are conscious or sentient. So what beings can suffer, what beings accumulate karma, and then I think a deeper question for our time now is what entities and beings are worthy of compassion? So in listening to the wonderful roundtable yesterday, I was very moved and inspired by the discussion, which I think was focusing very much on these questions, which I'll dive into throughout the course of my talk today. So again, in learning more about Buddhism and comparing it as well to other traditions that I've been inspired and impacted by, including Shintoism, indigenous animism, one thing that I thought was found very interesting was this idea that sentient beings with consciousness in the Buddhist framework include humans, animals, insects, perhaps in some Buddhist traditions, maybe plants, but not necessarily things like organisms like bacteria or yeast or fungi, for example. And so, again, I think there's this technical question around what is able to suffer in the context of Buddhism and thus be a subject to karma and also worthy of compassion. And so I think, Shintoism, I'm very, again, inspired by this idea of kami, the idea is that there are sacred spirits that can be present in almost any type of an object or entity, including tools and in rocks. And in indigenous animism, which again, I've also been very influenced by indigenous cultures around the world, there's a very deep idea around relationality, and that the weather, animals, quote unquote inanimate objects can also have animacy. And so I think these are really interesting frameworks to compare and to reflect upon, particularly when we consider synthetic biology. And again, depending on our definitions of life, of sentience, consciousness, there's very profound impact in our ethics. Right? This is very famous, of course, from Descartes, this idea of this animal machine, that animals are just robots, they're automaton. And so if animals are just robots and automaton, if they're just like chairs or objects, then we can throw them away, we can treat them poorly. And so this idea of animacy, I think, is very important also when we consider synthetic biology and these myriad synthetic life forms that are now being created, not only using biotechnologies, but also guided through computers and also artificial intelligence. I appreciate very much this quote from the Dalai Lama. If scientific analysis were conclusively to demonstrate certain claims in Buddhism to be false, then we must accept the findings of science and abandon those claims. So I think, in thinking about this panel and thinking about the future, our evolution and our ability to think deeply about what does life mean? What does sentience and consciousness mean? How do we define that? Are there new insights that allow us to evolve these definitions? I think are very critically important. And so returning to synthetic biology, this is, again, is a research domain that I grew up in. I've been at MIT almost my entire life, again, with my father being a professor. And for 20 years or so, synthetic biology has been advancing as a field, and it was really developed by engineers. It was developed by computer scientists, electrical engineers, mechanical engineers that wanted to apply engineering principles to biological systems. And I love this image. This is of a bacteria, of a microbe, but it has all of these circuits on it and so on. And so it's a very industrial set of metaphors thinking about life. DNA as code, for example, or cells as computers. And these are the metaphors that really are foundational to synthetic biology as a field. And critically important to synthetic biology is, of course, DNA. DNA as a molecule and nucleic acids in general, DNA or RNA, can be considered essential for biological life on planet Earth. And recombinant DNA is a set of technologies developed starting around in the 1960s that allowed researchers to copy, edit, cut and paste DNA. But really, it was the advent of DNA synthesis, I think, that we will reflect back on as a truly revolutionary technology in the history of humanity. And DNA synthesis involves using a machine called the DNA synthesizer that can generate completely synthetic, novel molecules of DNA that have never before existed in nature. So you can use a computer, or even today now artificial intelligence, to design brand new molecules that have never before existed and use these DNA synthesizers to create them and manufacture them. And we can manufacture DNA now, synthetic DNA, and build constructs of many, many scales, from individual genes, which could be hundreds of thousands of bases, to what are called genetic circuits, again, using these engineering metaphors that could have many genes in one piece of DNA, all the way up to hundreds of thousands of base pairs of DNA. And this is a field called minimal life, which I'll talk about in a moment. So these are the genomes of completely synthetic organisms. And in my own research, I've developed many different types of DNA synthesis technologies over the years, working on something called microfluidics or lab-on-a-chip technology. These are images of various devices I've developed with my research and collaborators over the years. And so these are very tiny factories almost that can produce and run different types of biological reactions in very, very tiny volumes, very small individual droplets, for example. And so using these types of technologies, over the years, I've worked on being able to miniaturize different types of synthetic biology processes, like synthesizing DNA or expressing proteins or culturing different types of cells. And as an example, for my own doctoral work, being able to use lab-on-a-chips systems to synthesize DNA and microfluidics. These are gene-length fragments. Later work involving the synthesis of genetic circuits, again, those larger pieces of DNA with many genes, and developing a novel hardware for that. Using 3D printing to manufacture different devices that, again, could be used for DNA synthesis. And using these synthetic DNA molecules, you can reprogram cells and treat them very much like factories to produce molecules that could be useful for humans. This could be therapeutics, medicines, chemicals, foods, flavorings, and so on. This is very much a very big promise of synthetic biology. I've collaborated with Professor Ron Weiss from MIT on making genetic circuits that could be used for cancer cell detection, and again, use these microfluidics as a way to detect cancer cells and produce many, many different types of genetic circuit designs. You can use these microorganisms that have been engineered with genetic circuits also to sense and respond to disease in the body. There's a whole field called cellular therapeutics, which has been advanced greatly in the many years. In my own research, we've used advanced 3D printing technology to make what are called artificial guts. So ways to explore the science of these microorganisms, the microbiome, the gut microbiome, but using it in a synthetic platform made through 3D printing. You may have already eaten some foods that come from this field of cellular agriculture, which is adjacent to synthetic biology. So foods that are made without using real animals, or clothes that are grown in a laboratory. This is work from my colleague Suzanne Lee with bacterially produced clothing, or using spider silk, for example, from colleagues at Bolt, which is a company in California making spider silk materials. And now, of course, with artificial intelligence, we are seeing incredibly explosive development in the design of proteins, being able to make not only predict protein structure, which is an incredibly previously an incredibly difficult area of science, but now to be able to predict the proteins of the entire known universe, and now to even being able to design completely new proteins that have never before existed. So again, as we think about this intersection between synthetic biology, computer science, artificial intelligence, we indeed are at a critical moment in the history of our civilization. One area of research that I think is very important is called synthetic cell research, and important relevant to this topic of Buddhism and the questions that I was posing earlier. Synthetic cells, this is a figure from my colleague Kate Adamala from the University of Minnesota. And the synthetic cell researchers, they're looking at being able to create, using from the parts and the components of a cell, but to be able to build a cell from scratch. So taking the components of a cell and putting them all together to make something that could be living. And there's, of course, a very big philosophical and technical debate in this field. Are synthetic cells actually alive? Going back to that question I posed at the beginning of this talk. And there's a number of criteria when we want to think about, could they be living? We think about, is there metabolism? Do the cells reproduce? Is there homeostasis? Do the cells evolve? Do they have autonomy? And my colleague, Kate, she likes to say, we don't know, right? There's no consensus agreement on what this definition of life is in the context of synthetic cells. So it's very hard to say if we can call them alive or not. And I will say, just to, my colleagues, they have a wonderful program called Build a Cell. I'm actually a part of this. And so anybody around the world, if you're interested in learning more about synthetic cells, you can participate in this program. So you can Google this, find this online. And I also have colleagues at the J. Craig Venter Institute, including my colleague and mentor, John Glass, who've been working on minimal cells, but looking at minimal life from the perspective of what are the minimal set of genes that are necessary to create a self-replicating organism. And so this is work from the Craig Venter Institute, where they were able to create a minimal organism, minimal life. And this paper was very interesting. They were able to use completely synthetic DNA. So again, using a computer to design DNA molecules, a DNA synthesizer using the AGCT components of DNA, building an entire genome. This is around 500,000 base pairs, and then putting it into an organism, and that organism actually could reproduce. And this was a real hallmark discovery and moment in science. But there's a wonderful quote, unexpectedly, this synthetic genome also contained 149 genes with unknown biological functions. Okay, so this idea of what is the minimal set of genes that are necessary for life is still being actively explored, and again, is one of the frontiers of science. So again, we can think about life from, again, a number of different perspectives, and this genetic one is also an important biological and scientific lens. But based on those considerations of what is living and what is not, I appreciate so much this work from colleague Hideo Iwasaki from Japan. Their group also looks at synthetic cell research, but really asks a philosophical question, a spiritual question. These cells, if they are living at some point, then might we not honor them like we would other living beings? And so they actually created a set of rituals and ways to commemorate the cells that are part of their research. And if you think about in the West, when we conduct scientific research, and certainly at MIT, we take bleach, we kill all the organisms, we pour it down the sink, and we don't think about it at all. It's a very utilitarian relationship that we have with these organisms. We think about them strictly from an experimental perspective and how they might be useful to humans. We don't necessarily think about them as sentient or having consciousness. And I think that's a very limiting view, ultimately. And so Again, I'm grateful to Iwasaki and his group for really pushing us to think about this context. And so again, for me, when I look at synthetic biology, I was raised in this field. I believe it's incredibly important and powerful, the types of technological innovations that are possible. And yet these metaphors, cells as computers, DNA as code, these metaphors consider living things in a way as inanimate, as not having consciousness or sentience. And yet when I'm in the natural world, this is a photograph that I took in the redwoods in California, near where I am now. I'm not thinking about cells as factory or these trees as lumber. I'm thinking about these trees as living beings, as ancestors, as entities and beings that are worthy of compassion and respect and even love. And this is a quote that I love from Robin Wall Kimmerer. Again, I've been very influenced and impacted by indigenous traditions and philosophies. And she's referring to animacy and the idea of living things in the English language being referred to as it. For example, when I refer to my mother is a she, a her. But if I am talking about the tree is an it. And so she says in English, we never refer to a member of our family or indeed to any person as it. That would be a profound act of disrespect. It robs a person of selfhood and kinship, reducing a person to a mere thing. So it is that in Potawatomi and other indigenous languages, we use the same words to address the living world as we use our family, because they are our family. And that for me was a very profound insight and feeling. Growing up in synthetic biology, where we look at cells as factories, computers, for me, deeply in my own spirit and being and belief, cells actually are living and are, I think, worthy of compassion at some level. And so I appreciate again very much this quote from Thich Nhat Hanh, we are the forest, we are the river, we are the bacteria in our intestines, we are not separate. I think this idea of life force on Earth and in the universe, that we are all connected, that there is a unity behind this all, I think is very resonant to me in my own journey, my own spiritual journey, and also in my own scientific and technological journey. And so, again, one of my great mentors, who has since passed away, LaDonna Bravebowl, who is a Native American in the United States, she spoke at a conference that I organized called the BioSummit. And this is my colleague and another one of my mentors, George Church, who is a very techno-utopian, Harvard and MIT synthetic biologist and researcher. And George was speaking about ways we could use organisms to address the climate crisis and to sequester carbon. And we could also leave planet Earth and travel to other planets if things got so bad on planet Earth. And LaDonna, she doesn't know who George is and was so furious after hearing him speak and said, who was that man that dare say we leave the Earth? Leaving the Earth is like leaving ourselves. And I thought it was so amazing, this tension between this techno-utopian, which I think represents many of the values and worldviews of MIT, Harvard, the West, and versus LaDonna, who looks at living beings on the Earth in a much more holistic, mutualistic way. And I started to teach a course at MIT called Ancient Future Technology, to start thinking about technologies like synthetic biology, artificial intelligence, but from a non-Western perspective, taking into account perspectives like Shintoism, indigenous perspectives, Buddhism, and then gratitude my colleagues Pratima and Andre for helping me launch this course. And to give you a sense of some of the topics in Ancient Future Technology, the course I teach, we look at storytelling, methods to secure indigenous guardianship of vital ecosystems, sacred technologies, technologies that can lead to feelings of awe and wonder, inspiration, natural collective intelligence, studying the collective intelligence of forests, of fungal networks, using synthetic biology also to de-extinct species for conservation applications. Some of you may have seen in the news recently this set of work done from Colossal on de-extincting the direwolf, so bringing back species that used to live on planet Earth and now in some biological way at least are represented now. Using artificial intelligence to decode animal communication. Or even using DNA synthesis, which I mentioned, but then taking those synthetic DNA molecules and putting them back inside of plants. So you could have a garden of plants that are storing artifacts that encode digital information in DNA. So again, we're living now in this time where the living world, artificial worlds, synthetic worlds, DNA, computation, they're all merging together in much more fluid and dynamic ways. And of course, artificial intelligence and ethics. And so the students in the course, you can look at ancientfuturetech.org to learn more. They all explore their own spiritual traditions and think about their own ancient future technologies. We also have social media, so you can look at some of the student work online. And we've been engaging in my lab and also research projects connected to these ancient future technology ideas. This idea of a project we've explored is called Living Artifacts. And again, thinking about different types of systems and objects where there could be, for example, microorganisms that we normally may not be aware of. Right now, I have trillions of microorganisms that are part of my body, but I'm not thinking and not necessarily aware or have consciousness of I'm not self-aware of these microorganisms. And so the idea of living artifacts was to create interfaces so we as humans could explore ideas of awareness. For example, awareness of the trillions of microorganisms. We might even be able to commune with them, to meditate with them, to share presence with them, or even to collaborate actively with them. And so there's a project my lab has been working on related to what are called biological cements. So these are wonderful materials that are actually living. They're living materials. You can take a type of bacteria called S. pasteurei and mix it with a number of reagents, including urea, a calcium carbonate source, and you can create a living material called a biological cement. And we've done some work, this is a more utilitarian research, to try to augment these biological materials with carbon capture capabilities. Or you can explore hybrid materials, bacterial and also fungal cements. So this is working with colleagues like Chris Voigt and Ren Romlin from the Biological Engineering Department at MIT, and also with colleagues at TO.org from Switzerland to enable anybody around the world to try to grow and make their own biocements. This is a topic that is very dear to my heart, which is democratizing these tools so anyone is able to participate and access these powerful tools. And I think for these biological cements, what's so interesting to me is that because they are living materials, if you think about the world that we inhabit now, in the room that I'm in the auditorium you are all in Taiwan, these materials are dead materials, right? They're not animate in a traditional way of thinking about animacy. And so here, these biological cements actually have living bacteria that are part of them. You need to actually water them and culture them and maintain stewardship to keep them alive. So it creates a different type of relationality with these objects. And so I'll conclude by sharing a little bit about a class that I teach called How to Grow Almost Anything. It's a synthetic biology course that I teach at MIT and Harvard, but also to students all around the world. And I'm inviting you, all of you here in Taiwan and part of this conference to come join us. It would be an honor to have you participate. And we teach our students all around the world and at MIT and Harvard the fundamentals of synthetic biology. At this class I teach with Professor George Church from Harvard, Joe Jacobson, and also many faculty from all around the world. And every single week, we teach the students a topic at the cutting edge of synthetic biology and also critically skills at the cutting edge of synthetic biology, how to design and edit your own DNA molecules, or how to, for example, design your own proteins. This is a very, very big area of research that I've mentioned, and the students learn all about the latest artificial intelligence tools, and they apply that to designing different types of proteins as a part of their own work. The students learn about designing and building larger DNA molecules that I've mentioned called genetic circuits. The students also explore minimal life. So one of our colleagues I mentioned, Kate Adamala, we do research with what are called cell lysates, which are some of these components of cells. And the students work both with synthetic minimal life experiments, but they also incorporate these cell lysates in a very practical way into, for example, textiles that could be frozen or freeze-dried and incorporated into materials on the body. We also teach a course on building genomes. So again, the pioneers of the synthetic cell work that I was describing, building that synthetic minimal genome, the leading experts come and teach a course on that, and the students learn all about synthetic life in that context. And we also spend a lot of time on lab automation. This is an increasingly important area of biology. I think is very much the future of how we are going to learn biology in the future is going to be mediated through robotics and also artificial intelligence. And I'll play a short video now from one of our students.
How to Grow Almost Anything is an MIT, Harvard, and global synthetic biology class.
Each week, students learn a key skill in bioengineering, ranging from topics like protein design to cell-free systems, bioproduction, and CRISPR.
This week, students learned about lab automation with the Opentron's liquid handling robots. Students programmed the robot to dispense fluorescent E. coli bacteria into the shape of a design that they created.
After growing on black agar plates for 24 hours, the living artwork glowed under UV light. Here are some of the amazing designs that the students created.
If you're interested in learning more about synthetic biology, check out htgaa.org.
And so the students, this is some of the artwork that they designed using the robotics, and all of these pictures are with engineered fluorescent bacteria. And so again, as I mentioned, the students come from every part of the world, which is why we would love for you in Taiwan and anyone that's in the audience to join our class. This past year, we had almost 1,300 students that were accepted to participate in the class globally from all around the world. And the course wouldn't be possible without all of the amazing instructors and TAs. I have almost 100 TAs that I work with to teach the course, again, at MIT and Harvard, and from every corner of the earth. I'm so grateful to work with all of my colleagues on teaching this course. And the class culminates in projects. So the students all work on a project that's very meaningful to them personally. And I'll give you some examples. This is a project from one of our students, Pat, who actually was a speaker at the Wooden Fish event last year. He made a device, a wearable device, that enables people, bacteria to produce molecules useful for space exploration. So this is Pat actually testing his prototype in zero-G. And again, this project that he took from the class ended up becoming a part of his master's thesis at MIT. We've had student, for example, Yishao Zheng from China, who had never been in a biology lab before. She ended up founding a biotechnology company making desktop biology labs. We have many architecture students that work on incorporating engineered organisms into architectural scale buildings, which has been very exciting. And there's so many different projects over the years, different types of biosensors that incorporate biological materials. I love this project, actually. This is a microbial kintsugi. So I'm using the kintsugi practice in Japan, but using biological cements to repair ceramics. Again, different types of sensors for detecting stress molecules in the body, low-cost modular biotechnological tools and instruments, organisms that can be incorporated into textiles that are responsive to temperature and pressure, computationally designed enzymes that can break down plastics in the environment, spider silk spacesuits, really just such an incredible array of projects that for me are so inspiring to see the creativity of all of the students from around the world. And again, you can go to the website to see more of the projects, but they're really incredible. And for me, I mentioned robotics, and again, this era that we're living in now. During the pandemic, the students could not be inside the wet lab, and so we had this very big problem. And so we taught the students Python, and we had them program robots from their dorm room or from wherever they were on planet Earth. And so the students were able to engage in the course by programming robotics that were held at MIT. So one of our TAs would be in lab with the robots, and the students could be anywhere. And so, for example, one of our classes, like on biological production, where the students were engineering organisms to produce what are called lycopene and beta-carotene. These are molecules that give color in vegetables, for example. And all of this was done remotely using robotics. And so we started thinking, we have a course called How to Grow Almost Anything, but it was really about enabling anybody around the world to participate. So growing almost anything, almost anywhere. And we started to really think about this vision of a global laboratory, a learning laboratory. And our students published, so we've published now a series of papers and work on this concept and set of ideas, which we're very proud of. And this is all, again, about access and enabling hundreds of millions of people, ideally, to be able to engage and learn about synthetic biology and to do it safely with an internet connection and a computer. And so this combination of what we call scalable learning networks, where there can be labs all around the world where students learn, but then also robotics, I think is a big part of the future of synthetic biology. And so I mentioned we had almost 1,300 students from around the world. We also had this year had physical nodes, How to Grow Almost Anything nodes, one in New York, one in San Francisco, one in Quito, Ecuador, one in Victoria and Ottawa, Canada, one in London, South Korea, in Seoul, and also in Tokyo in Japan. And again, we'd love to work with colleagues in Taipei to also set up a How to Grow Almost Anything node or a lab where the students could go in person. And we've also worked on ways for the entire network to collaborate on research together. So all of us as a global family and a global community can work together to solve problems. I won't get into the technical details, but last year we started a project working on what's called phage therapy, which is a different type of therapy to address antibiotic-resistant bacteria. And so the students all around the world design their own what are called lysis proteins. This is just a neat video showing a microfluidic system from our collaborators at the company Nuclera. Every single one of these pixels is a different DNA design from the students, and using the cell-free materials from the synthetic minimal cell, they're able to express these proteins all in this device. And so our students have been working on making these proteins all around the world. And this, to me, again, I think is a huge exciting part is global collaborative research. What can we do together? And what can we do together also powered by robots? So this is two of my colleagues, Tom Knight and Jason Kelly, two very important figures in the history of synthetic biology. They are founders of a company called Ginkgo Bioworks. And at Ginkgo Bioworks, they have very, very impressive automation systems called racks. There's basically something called a cloud laboratory, or robotic cloud lab. And this is very much analogous to early computers from the 1960s and many, many decades ago, where early computers might have been the size of an entire room. And now there's biological laboratories that are automatable, programmable, that have similar powers and capabilities. And so we're now collaborating and exploring the way that these cloud laboratories could be used for learning so that anyone around the world can interact with these cloud labs and have a very powerful learning experience with synthetic biology. And so for me, this idea of robotics, automation coupled with networks of people, I think is very powerful. And again, points to, I think, where we are in this historical moment in civilization. It's not just about synthetic biology and creating new living beings. It's doing it with automation technologies, very sophisticated robotics, and now artificial intelligence. It's a convergence of many, many important technologies. And that returns us, I think, to the very important ethical and spiritual questions that are being asked at this conference and that I mentioned earlier. As we build these synthetic entities, these are artificial intelligence entities, but also these synthetic cells, for example. How do we consider them as sentient or conscious beings, and might they have karma? I reflect on the observation of the Dalai Lama that as the science evolves, we must be thinking about how we consider and relate to these beings. Again, I'm grateful to, my Indigenous colleagues for their reflections on this idea of relationality, which I think is so important. And for me, I do believe in this moment in the history of our civilization that we do need to have a deeper, more mutualistic relationship with the natural world. And I think about this question of compassion very much, and I reflect again on Thich Nhat Hanh's remarks on interconnectedness. I think that as we advance as a field as synthetic biologists, we should treat living beings with greater respect and mutualism while we continue this very important research. And so I'll close by inviting all of you again to take How to Grow Almost Anything. To the Venerable Yifa, it would be an honor to have you come and lecture and speak about the importance of Buddhism and spirituality to all of these young synthetic biologists from all around the world. And so I thank you so very much again for your time and attention. Please reach out and contact me. I'm at David Sun Kong on all of the social media platforms, LinkedIn and so on. So please find me. I would love, love, love to continue this conversation. And again, this is a picture of my family and my sister and my mother. So 真的大家谢谢. I'm very, very proud to be here. And 我真的希望我可以跟你们在一起. So thank you all very much. 谢谢.